Okay, so I'm starting the second part of the lecture. And now I'm going to be looking at the origin of life, which is the handout, essentially chapter 3 of Meyer, and expanding on that, but also this other little handout that I gave you, titled The Origin of Life, is based on a book by Barton and Associates. The title is Just Evolution. So that's the reference on top there. And there are these 7 plausible states, the steps. These 7 steps could generate life organically on Earth with the matter and the energy that has existed on Earth over the millions and millions of years. Okay? So just to wrap up then this other origin, this origin of Earth is known as accretion. Accretion, which is a condensation phenomenon. So we're talking about 13.5 billion years ago was the explosion, the Big Bang, popularly known. And then we speed forward to about 10 billion years ago, with the formation of galaxies. Galaxies, what happens is here, as this author says, gravity starts winning. Gravity starts winning in the sense that matter is attracting more matter. That's the gravitational force, right? It's a pulling force. It's a pulling force in. And so as matter is condensing from gas to liquid, from liquid to solid, we start getting galaxies, which are formed by solar systems, otherwise known as planetary systems, because they contain planets. Not all suns apparently have planets, but a number of them do. We're discovering that increasingly suns have planets around them, which make them planetary systems. And these planets are opaque, as opposed to, in contrast to suns, which are luminous. You can say basically there are 2 types of bodies according to light emission or light absorption in the universe. Luminous bodies that radiate, that produce their own light, typically by a burning process, right? These are what we call the suns. They give off light. In the process, they also give off heat. But then there are other bodies that are opaque in the sense that they don't produce their own light. Rather, they absorb light, but they also reflect light. So when we look at the moon at night, whether it's a crescent little sliver of moon or a full moon, is the moon luminous or opaque? Luminous or opaque? The moon. Opaque. Right? But why, Father? I looked at it and it's bright, quite bright as could be, that full moon. What is it giving off? It's reflecting the light from the sun. So what happens is that the moon is off center, it's not on the same axis, right, of the Earth to the sun, and because it's off center over here, the sun is hitting it. So for the moon, when I see a full moon, it's daytime in the moon. It's daytime on the moon, and what I'm seeing is the daytime of the moon, which for me it's night because I'm on the backside of the Earth, so facing in relation to the sun. All right, but moons and planets are opaque bodies. We absorb energy and we also reflect light. We do give off heat from our internal core, which is hot, very hot. So to summarize all that, the formation of planets and therefore the formation of galaxies is known as accretion. Accretion, which is basically a condensation process. but massive condensation process that the typical result is planets, okay? That for us, meaning planet Earth, happened about 4.5 billion years ago. So that would put us about 9 billion years after the Big Bang, somewhere around here. Okay. So we pretty much began with the beginning of the galaxies. Makes sense? So we can land it on planet Earth, this little tiny speck that is going around the sun, which is embedded within the Milky Way galaxy, which is one of billions of galaxies that is out there. Mm-hmm. And so far, the only one that we know of with life, organic life. But when I talk about life, at least in this part of the course, I'm talking about organic, physical, physiological life of metabolism. So all that summarizes this little phrase here, the origin of Earth by accretion. That presupposes a Big Bang previously, about 4.5 billion years ago, BYA, billion years ago. In less than 1 billion years, and this, of course, accretion was a huge ball of red hot or almost white hot lava as gases were condensing into the molten liquid that is forms the inside of the Earth. Eventually, this ball that we call Earth gave off enough heat to start condensing further on the outer layer, which we call the crust, which forms the planet, the continents, right, and the supercontinents, and the plates that form the crust. They're known as tectonic plates. Tectonic plates, when they rub together, they cause earthquakes, right? Huge seismic stress. And these plates are moving around. And when plates merge against each other and they continue to push against each other, they rise and they form mountain ranges, like the Himalayas and the Rocky Mountains and the Alps and the Andes, all right? These are tectonic plates pushing against each other. So at the opposite end of the plate, they're receding from each other. But these plates are on top of magma, which is molten liquid, thick liquid rock, extremely hot, periodically comes out as lava through volcanoes or other cracks. Things had to get cooled off on Earth to have liquid water. The three basic states, solid, liquid, gas. We have water in all those three states on Earth, but the one that is necessary for life is the liquid state, the liquid phase. Why? Because life at the organic level is basically what? It's a bunch of chemical reactions that have some kind of order to them, and that order we call metabolism. And this metabolism is subdivided into two main aspects: the building up of molecules and the breaking down of molecules. So the building up of molecules is anabolism. Ana constructs and kata destroys. Catabolism is the breaking down. And metabolism is that constant flow of building up and breaking down organic molecules within our body in an orderly fashion that allows for cells to live, and clusters of cells form tissues, and clusters of tissues form organs, and clusters of organs form organ systems, and then several systems comes together forming an organism. All right? So the basic unit of life is going to be, that is stable, is going to be the cell at the organic level. Just like the basic unit of matter is the atom at a stable level, the basic unit of life is going to be the cell. So how can we get a cell? About 4 billion years ago, a little less than 4 billion years ago on Earth. from the organic perspective. You with me? Okay, evidence for the origin of life is this type of carbon called graphite, all right? Now, organic molecules. Organic molecules have to have at least two types of elements. Organic molecules. They have to have carbon. But what else do they have to have? Not just carbon, because, for example, CO2 is what? Carbon dioxide is not an organic molecule. Yes, hydrogen. They need hydrogen, okay? So basic elementary, this should have been an elementary school, so that's why you have to dig deep down in there and pull out that. We should have been reinforced at high school. But for an organic, for a molecule to be classified as an organic molecule, that molecule has to have at least these two elements. Typically, there are two other elements or three others that are associated with it, all right? But they don't have to be. And those are oxygen and nitrogen, and then other elements also, phosphorus, etc. Sulfur, yeah. But at least these two, this makes an organic molecule. Now, there are four types of organic molecules. Macromolecules, they're also known as macromolecules, according to their composition. Okay, and we'll see how these start making sense. And we'll get more into them again in the environmental course next summer. But basically we have what? If it has carbon and hydrogen, carbohydrates, right? The carbs, carbohydrates. And then we have also lipids. And lipids are fats, oils, waxes, greases. Lipids, they're typically insoluble in water. It's one main characteristic. And then the other ones are proteins. Proteins that do basically the work of that metabolism is done mostly by proteins, thousands of different proteins that we have in each cell, because many proteins are enzymes. Not all, others are complex that form hormones or steroids, etc. But all enzymes are proteins. Not all proteins are enzymes. But enzymes are the ones that speed up the chemical reactions in our body, again, metabolism. And finally, the last group of the of the organic molecules, which are essential for control and storage of information, nucleic acids, exactly, which are more recognized as DNA and RNA. Deoxynucleic acid, deoxyribonucleic acid, and ribonucleic acid. Or wherever you can punch it in, figure out the acronym. So just to say that these are the organic molecules, the groups or types of organic molecules, and all the bodies of all the organisms that are living on Earth are made up of combinations of these organic molecules. They tend to be large, including at least dozens, if not millions of atoms, right? So they tend to be large, and that's why they're called macromolecules, macromolecules. I'm just giving you some basic terminology here. Now they're still microscopic because they still fit within a cell, which is microscopic, but large in their context, in the sense that they're made up of at least dozens, if not millions of atoms. Like, for example, DNA, the DNA strand, chromatin, if I stretch it out, could be as long as 2 meters, 6 feet, inside a single cell, all right? So that is humongous. millions, in fact, billions, right? 3 billion base pairs in that DNA strand. Or we can get as small as a glucose molecule, 6 carbons, 6 oxygens, and 12 hydrogens in the dozens. Ammonia, well, actually, they're inorganic, NH3, no carbon, right? Speaking of ammonia and methane, which are the gases of the original atmosphere. So when we go back these 4 billion years ago, we go back to 4 billion years ago, what was the Earth like? It had a crust that was cool enough to have liquid water. All right, so what is the temperature of liquid water? Has to be a range from 0. Someone taught me how to make the little degree sign. Anyone remember? It's a control O. No, I messed it up. Anyone remember how to make the degree sign? The little circle? Control 0?
No, it's you press Alt and 0.
Oh, Alt 0. Alt 0. Not on mine. Maybe Control Alt? No. Okay, let me see. Then I have to go into subscript. This is the old way how I learn it. Insert symbol? Okay, let's try insert symbol. Okay, insert symbol. Where are you? All the way. Okay. And then now, yeah, now I need to find degree. Gotta hunt around. Usually it's around early on.
It has degrees Celsius.
Oh, yeah.
You saw it?
It was up a little, it was up in the...
Oh, where it was originally?
Yes.
Yeah.
Which was way down. Okay. It was around here, no? Higher. Maybe.
There it is.
Okay. That's Fahrenheit. I'm missing it. Okay, thank you. Okay. Insert. I should do it. Okay. Whoop, too many. So notation, right? The little circle above the number on the upper right is degree, and the C for Celsius, otherwise known as centigrade, right? Because the scale is from 0 to 100. Okay, so it's a decimal scale. 0 to 100 is a decimal scale. And liquid water is in the range from 0 to precisely 100. Okay, just copy and paste. All right. Consider that. Now, that is in centigrades, right? To be familiar, more or less, what is the range in the British system for Fahrenheit? At what temperature does water freeze in Fahrenheit? 32, right? 32. Oh, I had it there earlier. Let's go back to 104 degrees Fahrenheit. Thank you. And then boiling, yes, to 12, all right? So that's pretty hot for Fahrenheit, right? Because we're used to the thermostat being in Fahrenheit. I can I see it? Right click. Tell you what. I'll click that. Change that to that. Oh, okay. There's a little circle there. This is the range. We're more familiar with these temperatures because what's comfortable in here right now, more or less, in Fahrenheit? About 75, right? 70s. And when you want a loose translation between Fahrenheit and centigrade, about 75 Fahrenheit, obviously, is about 27 centigrade. So you invert the numbers, all right? More or less. 75 is 27, more or less, in that range. So it's comfortable range. 32 is very cold, right? Below 32, water freezes. Still water, but it's solid. Not available for metabolism. What happens to animals? They hibernate. They freeze, they slow down. 212 is way too hot. You ever get a vapor burn? Water vapor burn? It's dangerous. It can be a high degree burn on the skin, all right? So that cannot sustain life, generally. Then you get the extremophiles, the extremophile bacteria, which love extremes. But this is the range for life. And actually, the actual life, it's even more narrow than that. What's the body temperature, normal body temperature? Well, in Fahrenheit, again, we know it in Fahrenheit, 98.6, right? 98.6, more or less, for the thermometer. Fahrenheit cannot be 98 centigrade, right? Attention to detail, we'd be close to boiling. So what's that in centigrades? I think it's around 32, 34, 36, no? It's in the 30s. 98, somebody want to punch it in and look up what's body temperature in centigrades? It's in the 30s. That's the range that we need. Sorry, 37. Yeah, 37, right. 37 degrees centigrade is body temperature, which is pretty warm. Imagine what the body does. Out here, it's 75. Inside, it's 97. So in Fahrenheit, in centigrade, 27 to 37, 10 degrees centigrade, or 20 degrees Fahrenheit, just from my heart to the outside of my skin. That's a lot of very good insulation. Very good insulation is the tissue of the body, mostly done by the lipids, by the fat in our tissue, conjunctive tissue. So anyway, I wanted to talk briefly about the primitive atmosphere, because it was not this benevolent atmosphere that we have, which is a large percentage oxygen, but really the largest percentage of the gas in the air that we're breathing right now is what? It's actually nitrogen gas, right? About 3/4 of it, 70-something% is nitrogen gas. And we're inhaling and exhaling that. And how come we don't die? Because nitrogen gas is inert. It's what is called inert. It doesn't affect us. It just comes in and out of the lungs, and we don't use it, the gas, okay? So at normal temperature, at the standard temperature, nitrogen gas doesn't affect us. Oxygen we use, and CO2 we release. But the primitive atmosphere is not the current composition. The primitive atmosphere had mostly methane, right? And what was the other? Ammonia. And something else. Oh yes, well, there was water vapor, and those are the main chemicals, the main gases of the primitive atmosphere, mostly because of everything that was coming out of the earth, exuding from the earth, volcanic activity and the cooling down. It was a very toxic atmosphere that would be for us today. But those elements were the precursors of jumping to organic molecules. So those elements themselves, not elements, but molecules, compounds, were inorganic, all right? But the jump to organic was by a combination of these molecules. So again, let's list them here. We got these are all vapors, right? Vapors. The primitive atmosphere. All right, so water vapor is going to be H2O, then ammonia, NH3. In one molecule of water, how many atoms in one molecule of water? 3 atoms, right? How many hydrogens?
2.
How many oxygens? 1. All right? So just clear on the scientific notation for the elements. About ammonia has how many atoms total? Total 4 atoms. How many nitrogens? 1. The 1 is implied. When there's no number, 1. It's a subscript, just like the coefficient in algebra. You say A, well, it's 1A, right? Okay, and how many hydrogens? 3. So we're all clear on the scientific notation, on chemical notation. Now the other one is methane. NH or CH, sorry, CH. Right? How many atoms total? 4 plus 1, right? 5 atoms, 1 carbon, and 4 hydrogens. Okay. Are all these 3 molecules organic molecules?
No.
Which one is the one organic molecule?
No.
Yes, methane. Okay. These 2 are inorganic. Now, the idea is to come up with some building blocks. I told you that the working horse of the cell is what? The working horse of the cell is the proteins. The proteins are doing the work because they are mostly enzymes, okay? And also structurally and functionally, it's mostly proteins. So proteins are made up of amino acids. And amino acids are small organic compounds. How many amino acids naturally in nature? 20. Okay, we can move from here. And there's a table of amino acids. Some are attracted to water, some are repelled to water, hydrophilic, hydrophobic, some are charged, some are not charged. But you see they're relatively small organic molecules. The little zigzag represents what? Yes, at the point, at the bend, at the angle, but it's a skeleton, right? It's a skeleton, so it has a carbon skeleton. So you can count the angles. Here is 1 carbon, here's another carbon, that's it. So this is a 3 carbon molecule, right? About arginine here, 1, 2, 3, 4, 5. You see, I don't count the extension because that's a nitrogen here. And over here, there's an oxygen. But the angle represents a carbon. So this is a 5 carbon structure, 5 carbon skeleton. 1, 2, 3, 4, 5.
Is this organic?
No.
Like,
So-
no, meaning what they cover.
Yeah, organic compounds. Why are they organic? They have carbon. Everyone has a skeleton of carbon, right? Carbon, but they also have hydrogens, typically the OH, and then there are other hydrogens around. So as long as there's some hydrogen, it makes it an organic molecule. All right, so these are 20 amino acids in nature. They exist today. Building blocks, these amino acids coming together in a chain-like structure, more or less like the wagons of a train, more or less, they form proteins. Okay? I'll give you the simplified version. There are a couple of steps in between. But the point is that proteins are made up of amino acids, of which there are about 20. There are 20 in nature. And it's like if you're building a house, You're building a building, you're going to use blocks. Now you can use concrete blocks or you can use bricks. Those are two types of amino acids. One type of amino acid is block, another type is brick. You can use a different color brick, a different size brick, that's a different amino acid. But they're all building blocks for the structure that one is building. So the key issue here is really the first step. We have in the primitive atmosphere, let's go back 4 billion years ago with a face mask so that we can be in that methane atmosphere, okay? And let's see from ammonia, water vapor, and methane, if we can get organic molecules. How can we put those 3 types of compounds together to get some kind of organic molecule, some kind of amino acid? Okay, well, that was done in 1952 by a professor and his graduate student. Yes, the Miller-Urey experiment, right? Stanley Miller and Harold Urey. Who was the teacher and who was the student? Okay, Miller was a student, right? But because he designed the experiment and ran it, he gets the credit. In other words, he's the first name. In science, the PI, the principal investigator, the professor typically gets the last name, or the last on the list. The last on the list. So the Miller, yes, Miller-Urey should be in your notes somewhere. Miller-Urey experiment. Some people say, hooray for Miller. Okay. So that's the contraption that they designed. And with this contraption, it starts here, okay? And here, they had water, which they boil to make gas, to make vapor. Here goes water vapor. So they're adding water vapor in here, right? But in here also they added, there's a water vapor, then the CH4, which is methane, right? And then there is ammonia, NH3. Is that too little? And then there's, oh, I forgot, there's also hydrogen, which is highly explosive. So if you put a spark in there, if you put electrodes and run a charge, it's going to go whoop. It's going to give you an explosion, a charge, because hydrogen is highly explosive. Hydrogen gas. And then that was run through a cooling system and see what comes out in the precipitate. In this liquid here, the little soup, hopefully something organic would come out of these. You're trying to combine these 4 molecules, these 4 different compounds, into something else. Rearranging the atoms and the molecules, and they got some amino acids. I'm clicking too many things, sorry. Okay, they got some amino acids, I don't know exactly which, but I think that eventually they ran this enough that they got the bulk of the 20 amino acids, eventually, here. Yes, and even this one was criticized originally for something, and then it was tweaked. So this experiment has been repeated, but one of the points of scientific experiments is that they should be repeatable and get the same results. And it has been. Once they did that tweaking, it has been repeated. And the long and the short is that the scientists have gotten amino acids from the mixture of these different elements, of these different compounds. I keep saying elements. Of these different compounds. And these 4 compounds were the gases that were in the primitive atmosphere. And that's why this is plausible, a possibility that this could be done at the organic level without any direct divine intervention, as if some kind of a magic wand, let there be amino acids, okay? I say direct because indirectly it implies that these molecules and these atoms that make these molecules and the quarks that make those atoms obey certain laws, which is the background to the four fundamental forces. Why is it that positive repels positive? Who invented that? Right? Science can only tell us positive repels positive. But why? It's a force. In other words, we don't know. Okay. So my point here is that without any direct divine intervention done in the lab by human beings, the mixture of these four gases with a supply of energy and possibly also ultraviolet, which can alter the bonding between atoms. The result was amino acids, which are the first organic compounds necessary for making proteins. John, please.
What was the tweaking that you said was different the next time they repeated it?
I'm not familiar with the details. I used to be, and I confess that I've forgotten what it was, but there was some to the 52 experiment. There was some, and I think it had to do something with that this spark would not match exactly what would be happening. The spark they were attributing to lightning, for example, but perhaps the UV had more to do with it, all right? Also, I've heard that perhaps these primitive molecules did not occur so much at the surface of the ocean, but rather in deep, with ocean vents, with volcanic vents in deep ocean, which would be much more stable environments, temperature-wise, and different type of environments. So maybe the original environment was liquid instead of gas. That's been the controversy, okay? Because these ocean vents have essentially produced life. Well, but the bacteria had to be there to begin with. These vents, I'll show you, these deep ocean vents have no source of sunlight. And yet they have a whole ecosystem developed where the basic feeding stuff is bacteria that feeds on the sulfur and the heat that comes out of the vents. They metabolize the sulfur without light. Okay, it is fascinating. And then that creates a whole ecosystem of animals that chew on the bacteria, then other animals that chew on those animals, and so forth. No plants because there's no photosynthesis, because there's no light. It's a chemosynthesis, exactly. It's an alternative chemosynthesis, right? So number one here then, generating simple organic molecules from inorganic molecules. But there's kind of a little cheat sheet, if you will, because there was already this methane was there, which technically is organic. But the Earth produced that methane, so that's no problem, okay? It is a gas that was formed somewhere underneath, within the crust of the Earth, that was being expelled, was being burped out by the volcanoes and other cracks on the surface, on the crust. All right, but the point here in this number one is to get these organic molecules formed by natural phenomena of mixing of chemicals with energy, replicating or imitating the primitive atmosphere to the best of our knowledge today, going back about 4 billion years ago. Next step. Then there is something called chemical evolution. In other words, molecules also evolve with time. Now, the key factor here is going to be this long periods of time, because we're going to start counting in thousands and in hundreds of thousands and in millions of years. So over long periods of time, those isotopes change, the molecular structures change slightly, and can give rise to different shapes, different configurations of the molecules. We're already in the organic molecule world, if you will, the organic molecule world, which is the precursor to jumping into metabolism, because these organic molecules are going to be the ones that will metabolize and be metabolized. The metabolizers and the metabolizees are going to be organic molecules. So from the simple, we get to complex mostly by polymerization, by joining of those various wagons into some kind of a train. Some trains are going to be longer, some shorter, but now we got polymers. We're going toward the macromolecules, the macromolecules. That can be done by adding ultraviolet radiation, which does cross-linking. In other words, how do you connect one wagon to the next wagon? That link at the molecular level can be done with ultraviolet radiation, because that's precisely what ultraviolet, the mutation that it causes on the DNA, it alters the shape of the DNA molecule, because it messes around with the links between atoms, all right? And it can distort the shape of the molecule. It can change the links, and it can cause what is called cross-linking. Atoms that were not joined will be joined and crossed over to the adjacent molecule. The point is that by adding ultraviolet radiation to some molecules, they can be linked together to form a macromolecule, to form that train. So that would be the step 2. And there was plenty of UV around in the primitive atmosphere because there were no clouds and there was no ozone and other molecules to block that UV. Third step, another crucial step, this would be like minimal steps to get to a possible organic cell that is living and working and metabolizing. The next step is self-replication. And we get these molecules to self-replicate. In other words, to make a duplicate of themselves. Okay? What do we mean by polymers? The first simple polymers are chains of the same molecule repeated several times, maybe a few times, maybe a few dozen times, all right? How do these molecules get to duplicate themselves? They have like a blueprint. We have some hint of that in ribozymes, because ribozymes are little organelles inside cells that have RNA and protein, and it's a combination of RNA and protein. And these ribozymes are the ones that duplicate the DNA precisely. And they read the DNA molecule and they make a duplicate of it. Yes. Yeah, well, first, yes. You get the replication. So one thing is replication, which is duplication. And the other one thing is transcription, translation, where you get a protein out of it. All right? Again, don't be lost with the terminology. I'll come back to it in detail, but just in the big picture for now, this self-replication, it can happen by cycles of hydration and dehydration. Hydration and dehydration. They say that DNA, for example, is very wet. What does it mean that DNA is very wet? Well, I told you it's a macromolecule. It's a huge, long molecule that is very coiled and super coiled into the cell. But the DNA itself is very charged on the outside, so it attracts water. It has like a coat of water, all right? And water is polar. Water sticks because it's like a little tiny magnet. And so when a molecule is wet, it means that it's coated with water. Technically, it's called that the molecule is hydrated. It's hydrated, it's coated with water molecules. But then with heat, that electromagnetic bond, that magnetic bond breaks because you're adding heat to the molecule, and then the water molecules vaporize and they let go of that other macromolecule. That's a dehydration process. The water is lost. There are cycles, there can be cycles. How can we have cycles of hydration and dehydration? By adding or taking off heat. How do we add and take off heat on Earth, in nature? In 1 year. Well, day and night is one cycle, that's a daily cycle, but also a yearly cycle, because our translation around the sun is not a circular translation, it's elliptical, all right? And so we have seasons. There's a time that is warmer, there's a time that is cooler, which actually has more to do with the angle of the Earth to the sun. But my point is that we have seasons. Sometimes it's cooler on Earth, sometimes it's warmer. And it also depends where you are on Earth, closer to the equator, less variation, toward the pole, more variation. Okay, but my point is that we can get, with time, with cycling, we can get hot, cold, we can get cycling at the molecular level, and we can get hydration and dehydration. And when that is repeated periodically, time and again, hydration, dehydration, hydration, dehydration, that could produce copying, could force these molecules or encourage these molecules to duplicate themselves. So the duplication of these polymers has been associated with hydration and dehydration cycles, with cooling and heating of the atmosphere and the surface of the Earth. Into the fourth level. The fourth level is another critical level, compartmentalization. Big word, but we understand the meaning. A perfect example of compartmentalization is our closet at home. We got stuff in there, right? And sometimes that stuff spills out. But it should stay in the closet. Is the compartment? Oh, I need the, no wonder, I need the other life. Give me a minute here, so I pull up same handout in the big picture. Yes, compartments. Our home hopefully has a living room, a kitchen, a dining room, a bathroom, and a garage, and closets, you know. Different activities, different functions occur within these different structures. The same with the cell, right? The nucleus has a purpose, which is different from the cytoplasm because the information has to be stored and kept safe within the innermost part of that cell, which is the nucleus. Then outside of the nucleus, but still within the cell, metabolism occurs. All kinds of enzymes are doing activity, and those enzymes could break down the DNA. But we need to store the DNA in that closet where it's safe. So there are compartments. These are all organic compartments. These are made by membranes. How do we make a compartment in a cell? And the cell itself is a compartment by membranes. How do we make membranes? Okay, then we have to get into the polarity of these macromolecules. We're already at the level of macromolecules. Already we have some lipids, some proteins around, okay? And some carbohydrates also. But let's stick with lipids and proteins for now. Lipids, I told you, were what? Examples? Yes, cholesterol. So greases, fats, oils, waxes. Yes? Do they mix in water? They don't mix in water, right? That's a common characteristic of lipids. They are hydrophobic. They hate water. Water, phobic, so phobia, hate them. Whereas other molecules, that's because they're not charged for that water to coat them. They don't become hydrated, right? They don't become coated by water, so they're hydrophobic. They tend to move away from water. So if I I have a little dish of water and I drop some lipid, and the lipid is going to stay within itself, all right? But if that lipid gets close to the edge, that lipid is going to stick to the edge and try to go away from the water, right? So continue to stay away from the water onto the edge. Whereas proteins can be either hydrophobic or hydrophilic or amphipathic, meaning that they have both. They have one end that is likes water, the other end doesn't like water. All right, amphipathic, I have the word somewhere in here. Anyone see it? That's basic for, yeah, here it is. Okay. Amphipathic. Pathic is that it has some pathos, some kind of like or dislike. A pathos, either a like, a positive or negative passion, right? So it's either a love or a hate. Amphi, both. Like amphibian, both land and water. Okay, amphipathic is that it has a passion for both, for loving water and for hating water. How can you have that on the same molecule? Well, first of all, it's a polymer, it's a long molecule. One end, likes water, it's charged, has a charge, so the water can stick to it. The other end doesn't have a charge, so it's repelled to water. So I have, yes, so that's where we get the membrane, the bilipid, the bilayer, the lipid bilayer, okay? So let's look at the cell membrane in detail for a moment. Called phospholipid bilayer. And that's what we get. Let's start with a simple example. This one. All right, you see these yellow balls, these little legs. Okay, these are lipids. And you notice that the head part, the ball, yellow ball, is facing, this would be water, and this would be water over here too. So the yellow ball is charged, positive or negative charge, which means that the water will tend to stick to it. Okay? Because water molecules are like little magnets. They have a positive charge on one side and negative charge on the other. So they're looking for charged surfaces. So this yellow is going to be attracted to the water. It's going to be facing the water layer. Here's water, excuse me, and here's water. But the legs of the phospholipid, okay, are hydrophobic. They don't like water. So they're going to hide, they're going to try to be away from water. And because they're neutral, they have no problem in facing each other because they're not repelled. They're not attracted either, but what's holding them together is the water on each side. And so these molecules are amphipathic, and the hydrophobic end of the molecule is on the inside. The hydrophilic side is on the outside. The hydrophilic end is on the outside. And that's how we end up with a bilayer. So it's a bilayer, they're facing each other, and the outside is charged so that water likes it there. And that water is keeping water outside the membrane, water inside the membrane is keeping that membrane together. Okay, so this is what is known as a bilayer, which would be where, fourth or fifth step? Okay, fourth step, because when you start getting these bilayers around, these bilayers in water can form like little balls, little spheres that are called protobionts, because they are the precursors. So in here somewhere.
Like microspheres, right?
Yes, here we go, protobionts, because they are the precursors, proto of bio, of life. They are the precursors of living cells, all right? And these are essentially little balls or spheres that are surrounded by a bilayer. But then what do you have inside? What do you have outside? Water. What do you have inside? Water. But what if on the inside, in addition to water, some other proteins get trapped in there? And those proteins start interacting with each other. They could possibly start metabolizing. They could start interacting with each other and either breaking down some substances or building up some substances. Then that's metabolism. And that's why when you get these little protobionts, these little spheres, these bubbles forming conceptually, they could lead to the precursor of a cell, all right? Again, we're at the very microscopic level here for sure, but possible. These could enclose particularly proteins, but also lipids and carbohydrates and nucleic acids. And these could evolve into what are known as protobionts. In other words, this is hypothetical, but it's possible organically to happen. you have to keep adding on top of these are layers. On top of this compartmentalization, there's also cycling going on of cooling and heating, which these molecules are going to react differently. Lipids expand and so forth with heat, and to the point that if you heat them high enough, you can actually get them dissolved temporarily into water. And it's also going to have the continuous UV action and so forth. So you have to keep adding the other abiotic, the other factors, the natural factors that are influencing or acting upon these reactions. The next step that is necessary, so I'm calling linking genotype with phenotype. The genotype is the information, the chemical information, the information that is encoded in a chemical molecule. That's the genotype. We know them as the genes today in our DNA. But we also know that possibly the early world, the early genetic world, was not a DNA world, but what has been known as an RNA world. An RNA world. In other words, the early information molecule could have been an RNA, which is a simpler DNA. It's not a double helix, it's a single strand. It would be coiled, right? But it's a single strand because it's only ribonucleic acid. It's not deoxyribonucleic acid. But also that single strand, because it does have the bases, and again, we'll cover in more detail later, those bases tend to attract each other. So the RNA tends to bend upon itself and stabilize to form base pairing. And that's called hairpinning, hairpin, all right? It looks like the woman's hairpin on the hair, more or less. But the RNA basically folds upon itself to stabilize the bases so that all those hydrogen bonds are not exposed out there, all right? That stabilizes, and that could be a precursor for a DNA molecule because now that what was originally a long strand of nucleic acids maintaining information, encoded information, has now doubled upon itself, pairing up, adenine with thymine, cytosine with guanine, the double pairs that we'll look at later, but eventually stabilizing the molecule and the precursor of the double helix of the DNA, the stable DNA, which is the basis of all organic life. Every single organism of those 2 million organisms, or 2 million species that have been classified today so far, they all have DNA. Some have RNA like viruses, but basically they all have a DNA as the genetic material, what is passed on from one generation to the next in the tiny molecule or the gametes, or by splitting and so forth, but it's the genetic material, the information of the whole species, of the inheritance, is in DNA. That's why we say that DNA is a universal inheritance molecule. But the precursor could have been RNA. Another indirect evidence, if you will, of possibly the early genetic code being an RNA world is that there are many different types of RNA today. In contrast to DNA, which is essentially one, the 3 configurations of the DNA molecule, but essentially it's one inheritance molecule, DNA. RNA tend to be smaller and in different types of configurations that is not a double helix, okay? There are things such as messenger RNA, transfer RNA, ribosomal RNA. I'm just indicating the lowercase letter here, okay? Others, short SNP RNA, etc. RNA interference. So there are many different types of RNA molecules within cells today, a great variety. So just like at the living level, when we saw different varieties of species that are similar, right? There's a brown bear, there's a black bear, there's a grizzly bear, and there's a panda bear. Well, but they're all bears. It's pointing toward a common, original, common ancestral bear. Well, similar, if there's so many different types of RNAs, it's pointing to one common primitive original RNA molecule. That's why they talk about this RNA world that makes sense before the current DNA world. So these are inferences because we have, what's the axis? Well, the axis is mostly the fossil record, right? But that has to be hard tissue that has been mineralized. No soft tissue is preserved for billions of years like this. Okay, more details in how to get the actual 4 bases of the DNA today, because it turns out the RNA doesn't have thymine, it has uracil. But if you methylate uracil, it's a chemical process, you get a thymine. So that would be adenine, guanine, thymine, and cytosine are the 4 nucleic acids that make DNA. We'll get to it in more detail in a couple of semesters, okay? Just a big picture. What I'm saying here in the step 6 is that it is, I'm sorry, I was in 5. To link the genotype with the phenotype. In other words, a macromolecule that has a chain can store information. Just like the different wagons of a train, some wagons are carrying wheat, some wagons are carrying rice, some are carrying cattle, etc. They are storing information in those wagons, right? How to translate that into some expression? That's what I mean by phenotype. Phenotype is expressing the code, the genetic code. So to give you an analogy in non-chemical terms, let's say, well, I'm actually reading from this page now, right? Let's say that these words, when I say linking, that has a particular meaning. And linking is different from apple. An apple is different from soccer, all right? Each one of those words has a different meaning, but they're all made up of letters. And some letters, I only have 26 letters to work with. So the different combination of those letters give me words with different meaning. But I only have the playground of the toys that I have is these 26 letters, okay? So the 26 letters is like the genotype. The formation, the information is there. How I use those letters will be the expression, the word is the phenotype. So how I combine those letters together will give me one word or another, and that is the expression of those letters in that particular sequence. That's the phenotype, broadly speaking. If we translate that into chemical terms today, the genotype is in the DNA molecule, which is super wound up into, like you take a rubber band and wound it up like that into the nucleus, right? That's the genotype. The information is stored in there chemically. The phenotype is going to do what the proteins do with that information. What the proteins make or break down outside of the nucleus, but still inside the cell in what is known as the cytoplasm of the cell. So maybe at this point it would be good to show a cell, since we're getting close to one here. Biologically, hopefully we'll give you some images. Yeah. Okay. Here's a simple model. You see that it's membranes within membranes within membranes, all right? Let's say the DNA is in here, and what's enveloping that DNA? It's a membrane, which is going to be a phospholipid bilayer. This is a double membrane of the nucleus, all right, which of the DNA, yes, and it's keeping that DNA in there so it doesn't move around outside the cytoplasm because it's going to be digested outside of the nucleus. So it's protected. This is one membrane, so the precursor to this could have been one of these protobionts. But it turns out here's another membrane. Here's another little ball, sphere, all right? These are known as lysosomes, all right? Soma, body, okay? Lyso, they lyse, they break down. So what's inside here, these lysosomes? Inside these lysosomes are lysozymes. Lysozymes are digestive enzymes. So if this little ball breaks, this cell is going to be digested, it's going to be eaten out. It's like adding pepsin to it, right? Pepsin, which we produce in the stomach. Pepsin digests what? Protein. We eat protein. We can swallow protein whole. We can swallow beef whole. That's why we can eat raw beef and raw protein, meat. It will be digested in the stomach by an enzyme called pepsin. Okay, so if this lysozyme breaks, the cell gets digested. So it better be enclosed in this membrane. But this, I told you that lysosome contains lysozymes, and lysozyme is a type of enzyme, and that enzyme is a type of protein. So other proteins made that protein, but when they're making it, they make it very careful, and they make it enclosed inside these other little membranes so it doesn't leak out until it's time to leak out. All right? And so all these are membranes. Yeah, this is known as the endoplasmic reticulum, and it folds out of the nucleus, and it gives structure to the whole cell. These are also membranes. This is known as the Golgi body or the Golgi apparatus. Anyone recognize this? Yeah, mitochondrion. All right, here it is, mitochondrion. Also a double layer. There's another layer. There's an inner layer that is corrugated. This is the energy factory of the cell. Okay, energy factory. If it were a plant cell, it would also have another huge, well, something similar to the mitochondria, but they're called chloroplasts. Those chloroplasts have chlorophyll, and they do the photosynthesis. So cells are full of these little structures that are called organelles. Okay? What do our organs do in our body? Our organs perform functions, right? The liver filters out the toxins in the blood. The brain thinks, etc. So different organs of our body have different functions. The stomach doesn't do what the pancreas does. Well, something similar, actually. It's involving digestion. Bad example. Doesn't do what the lungs do, all right? And the lungs do what the heart does. But different organs do different functions in the body. By analogy, these structures do different functions in the cell, and that's why they're called organelles. Organelles. They're like little organs for the cell. The brain of the cell, if you will, is going to be what? The nucleus, because it stores the information and it controls, by that information, it controls the activity of the cell. But the activity of the cell itself is what I mean by metabolism, at the organic biochemical level, okay? And that metabolism occurs within the soup that is mostly water. It's a jelly-like water, very hydrated, and that's called exactly this cytoplasm, the cytoplasm. So basic biology from high school and maybe even elementary school, just to put everyone on the same page. We should be around number 6 of the origin of life. Okay, the genetic code, that's what I'm saying, is that this RNA, if it was an RNA, well, to begin with, just like amino acids put together make proteins, nucleic acids put together make RNA or DNA. Okay, so you get amino acids for proteins, nucleic acids for DNA or RNA. The translation, not going to belabor it any longer. It's possible in the lab to go from RNA to DNA. Finally, the 7th step is to have several characteristics. Number 1, DNA as the information molecule. Number 2, that DNA compartmentalize within a nucleus so that it's protected from the digestive enzymes of the cytoplasm. Then a further membrane to contain the cytoplasm itself, so that each cell, I can say this is a cell, and outside of it is either another cell or no cell. But the cell is compartmentalized, just like our body is compartmentalized by what system? The integumentary system, otherwise known as the skin. Epidermis, dermis, and then there's the adipose tissue and so forth, but the epidermis and dermis is a whole system that contains our bodies, all right? Okay, so DNA as the information molecule, compartmentalized. able to replicate, to duplicate, and to metabolize. Basically, that would give a cell, a living cell. Individual steps, all these 7 steps have been done in the lab individually. The trick now is to put them together in a sequence. And to my knowledge, to date, I don't know of any lab that has managed to go from 1 to 7, okay? But each individual step has been done in the lab with the material, with the precursor to get the end result. The trick now is to put them all together to get the living cell, starting with gases and energy. But it's possible. In other words, there is an organic, biological, physiological, physical, and chemical explanation for going from these gases in the primitive atmosphere eventually to a living metabolizing cell without any direct divine intervention. We as Catholics have no problem with that because our question of God is further back to a previous origin, if you will, at the level of principles or laws, okay? But not at the level of a magic wand pulling rabbits out of a hat. That's a God we don't believe in. Okay? So what would be the minimal characteristics of a living cell? That it metabolizes, like I said. Building up is called anabolism. And breaking down, like digestion, is called catabolism. Then that it would have, these are characteristics of the DNA, how that information is either duplicated, which is called replication. The DNA information, the DNA molecule duplicates with a machinery, all right, with a chemical machinery, that's called replication. Or the other alternative for DNA is to send out information from the nucleus out into the cytoplasm for the proteins to do something, to get up and do some work, right? To metabolize. That information is in the 2-step process. The first step is called transcription. In other words, pass from DNA to RNA, and then the RNA is sent out into the cytoplasm. And then that RNA is read, is translated into a protein molecule, a particular protein molecule that will do a particular work. So that double step is called transcription, translation. Yes?
That would be, suppose RNA came first.
Yes.
So when you go into the transcription, when it reads the RNA, how would it happen first?
Again, with the lysozyme that you, I'm sorry, not the, with the ribozyme, which the ribozyme is a complex of RNA and protein. And it has that 2 functions. The RNA has the information part, and the protein does the work. The ribozyme, right? They're in the ribosomes, and they're the ones who make precisely the proteins. Okay, but their DNA, it's an RNA protein complex, right? The ribozyme. That was he gave the Nobel Prize to that fellow in University of Colorado Boulder. I forget who discovered the ribozyme and the complex that it is. A combination between remember has subunits of the machinery that reads the transcript, translates it into a protein, right? Putting amino acid by amino acid. That complex is a ribozyme, meaning that it has RNA and it has protein. It's a large, complex macromolecule. And you can see there is genotype and phenotype because RNA has the capacity to store information. In this case, the information is coming from the messenger RNA in the nucleus, from the nucleus, okay? But that RNA in the ribozyme has the capacity for storing information. It's not being used currently in this function, but it has capacity. I think in fact, actually, it codes for part of the ribosome. And then the protein.
From the DNA though, like the messenger from DNA.
No, it's different. This is rRNA, ribosomal RNA, okay? Little r, small r, rRNA. That's the ribosomal RNA, ribosomal RNA. That's the one that makes up the ribosome complex. That together with protein, they're able to read the messenger, and then the transfer RNA is the one that is bringing the amino acids one by one to lock them into the triplet codon, all right, that whole machinery.
I forgot the name of that right now.
Yeah. Okay, but it's a whole complex, you know. In other words, this occurs physically, chemically, organically in the cell, and I would say it's automatic in the sense that if you put these elements together, it will run by itself. It would run, it will read the DNA, it will read the RNA, it will make the protein. The particular code that you give it, the particular sequence of nucleic acids will produce a particular sequence of amino acids. That's also known as the genetic code. You can tell I'm too excited about this. It just gets me into the trouble of the detail. And I have difficulty explaining it, but this was deciphered.
What is it? Yeah, the codon and the antigen.
Exactly.
Yeah. And then it reads.
Yes.
Something with column.
This is what we're talking about. We're talking about the genetic code. I told you earlier that there were 4 bases or 4 nucleic acids, right? I told you it was adenine, guanine, cytosine, and this is a translation of thymine, because thymine in DNA becomes uracil in RNA. Don't worry about the details, but if we do a table of this, we put nucleic acids, which are only 4, and nucleic acids, and we do combinations in 3s, we have these 4 possible combinations each box will have 4 possible combinations of the nucleic acids, and these are what codes for the amino acids. So what is shown in blue here mostly, and then green, blue and green, are the amino acids, the ones that make the proteins, okay? So don't confuse nucleic acids with amino acids. Nucleic acids are the building blocks of RNA and DNA. Amino acids are the building blocks of proteins, okay? The 20 proteins are here, are illustrated, the 20 amino acids are illustrated, mostly blue, 1 green, methionine, and tryptophan here in black. But there are only 20. But if you count these, there are a total of 64 of these combinations. There are a total of 64. Okay, so that's what is called redundancy. In other words, these 4 triple codes code for a single amino acid, serine. Okay, these 4 combinations, they all code for this one, because if you notice from the code, the one that is important is the first 2, which are the same. The third on the code, on the genetic code, the third base, in this particular case can be any one of the 4. It's called a wobble, don't worry about it, but this one is irrelevant as long as it has 3, but the first 2 are the ones that code for that particular amino acid. In this case, the wobble is only 2 wobble, uracil or cytosine, but the first 2 have to be always the same. Okay, so the first 2 are the one that count, and the third triplet on the code can wobble around.
What are the different colors? Are they
Colors, because blue is for amino acids, except for the start codon. So every messenger RNA, when it has AUG, that's a code for methionine. And methionine is the one that starts the code. That starts the read of the code. From methionine, from AUG further downstream, it's all sets of 3s. Those are 3 codes, sets of 3s, all together, all right? Because it's a whole string. You don't know when it starts and when it ends, except if you know the starting one, then everything else after that follows in groups of 3s, sets of 3s. And so methionine is the one that starts the code. It's called the starting codon, okay? Which goes for, is AUG on the RNA, which would be on the DNA. What would be the code for AUG? Well, I said that uracil is replaced for thymine, okay, so it would be ATG, but it's actually the complement. It will be the complement, all right, because it's reading it off. So it would be A goes with T, so it would be T. T goes with A, so it would be A, and G goes with C, so it would be TAC. On the DNA code, it would be TAC to code for AUG on the RNA, on the messenger RNA.
The chemist is confused by the bio.
Yeah, yes, exactly. So you have to switch worlds back and
So
forth
the
like
DNA
that.
has this
The
is
code.
actual RNA
And this is the
comes off
transcript of the code,
Got it.
exactly. Those are the technical terms, okay? The DNA has the code, and the DNA contains that code. The DNA does not go out into the cytoplasm. It's extremely dangerous. It's going to get digested out there. The DNA sends a copy of itself in a single strand.
And that's what
That's the
transcription-
messenger. That's the messenger
Got it.
transcript.
And the
Yes.
translation when the RNA leaves.
The translation is the last step, is translating the code on the RNA to a protein. To a protein, going from RNA to protein. Okay? So you have transcription from DNA to RNA and translation from RNA to protein. And then the other colors, so you got all blues are 18 of the 20 amino acids. Then the 19th, or the first one, is methionine in green because it's a go. And then tryptophan here in black because that's also a stop. That's where it ends. In other words, you got to start it somewhere, but you can get again that you have to end that train somewhere. That train cannot go on forever, okay? And it ends with UAG or UGG. Those are stop codons. Mm-hmm. Or this one UGG codes for
So it'll
tryptophan.
basically start transcripting. Once it finds a methionine,
Yes.
they'll say, "Hey, listen to me." This is
Yes,
important.
exactly.
Until it reaches the tryptophan.
Everything behind me is important. It's crucial.
Exactly.
Okay?
The
Mhm.
stop, the stop, are they all tryptophan?
No,
Oh, okay.
only UGG is tryptophan. Actually,
got
this one
one.
actually codes for tryptophan,
Right.
but the other three
It just
code for
stops.
stop. Yeah. Mhm. And I think, yeah, that should be the only one for tryptophan. Yeah, so tryptophan is unique here. So if there is a mutation on G for A, that's going to cause a big mutation in that particular protein because it will not have tryptophan. It will be cut off prematurely. It will probably make for a non-functional protein. Or it could be an alternate function, but that could be a genetic defect there, just a single base switch. Okay, let me go back. Where am I? I'm on the 7 steps of possibility of origin of life at the organic level. Okay. So if the DNA can reproduce and if the cytoplasm can metabolize, one can envision that one of these metabolic processes is to duplicate the whole cell. Segment by segment in an organic fashion, in a method, in a step-by-step process, which we call mitosis, cell division, okay? Which happens, we observe it in nature all the time. It's happening us in our bodies right now in the millions of cells that are duplicating by mitosis. So if that occurs, then that's one of the key points for life that can reproduce itself with more or less fidelity, all right? And then within that cell, that there will be some variation which in the long term could lead to speciation, especially if there is geographic isolation or local isolation. The variants will start emphasizing one aspect, either longer wings or longer tails or longer legs, and could lead eventually to speciation. This is still at the conceptual level, but what I'm saying is that these concepts are realistic. Okay, they can happen. So within an open system, why is the Earth an open system? You ever have aquariums at home? You have aquarium, freshwater, saltwater, you know, the simplest aquarium is just a glass ball with water and a goldfish. The more complex system would be a saltwater aquarium where you have anemones and you have corals and you have animals, plants, you have algae and so forth. Okay? You can have, obviously one is much more complex, but basically an aquarium is a closed system. It's enclosed within that glass physically. You have to feed it and so forth. It's a closed system. The ocean, we call an open system because there's interaction with the atmosphere. There's river runoff. There's even water, salt water that is percolating through the bottom of the ocean, through the bottom, the abyssal zone, into the earth, and cycling there and picking up nutrients and coming back up again. So it's a dynamic open system, all right? Over a long period of time, and a different scale that we're used to, but it's an open system. But the earth itself is an open system. And why is the earth an open system? Think of energy flow, the energy that flows through the earth. We get a lot of what kind of energy? That makes day and night. Solar light. Light energy comes into the system, to the earth. But what do we give off also? Mostly, what do we give to the universe that we don't keep here? Thank God, heat. Otherwise, we'd be boiling by now, right? So we give off heat. We radiate heat out, which dissipates into the universe. That heat is unavailable for work. That's what is called entropy. Entropy is energy that is unavailable for work. There has to be a balance because the universe was already created and there's no more material creation going on, all right? The sum of the matter and the energy in the universe is already contained within the system of what we call the universe. But each individual part of the system can have a flow of matter and energy coming in and out, right? And that makes that particular part of the system an open system, a dynamic system. And the earth is that kind of system, especially at the energy level, whereby we receive sunlight all the time. Half of the earth is always receiving sunlight, okay? But the whole earth is also radiating out heat. So there's an energy transformation. Otherwise, if we just receive light and light, at some point we become luminous, right? But we're opaque. So there has to be a transformation of energy. What is that transformation of energy? Well, typically what happens when light hits the surface, two things happen. Some of the light is bounced off, and we get a reflection like the moon at night. Some of the light is absorbed and changed. And some of the light changes into heat, all right? That is the heat that is given off by the earth, part of it. But the other part is also internal heat from our own because we are cooling down. The earth, is cooling down little by little. Remember, 4.5 billion years ago, we used to be a ball of fire that started cooling, right? The accretion. And cooling enough to have a crust that is solid. But the center, the mantle of the earth is liquid, molten, yes, thick, but liquid. And that's why the crust is moving a little bit. Because it's on top is these floaters, these rafts that are on top of this molten lava. And the molten lava is having convection currents, just like any liquid is circulating and moving. That makes the surface move. But the very core of the earth is back to solid. It's mostly iron because it has to maintain the gravity, right? Otherwise we would just explode. The gravity of the iron core, which is solid, mostly iron element, and it is white hot. Beyond red hot, what is white hot? Remember the incandescent bulbs before neon and before the LEDs, all right? There were the incandescent bulbs, still have incandescent bulbs at home. They get very hot. When you turn off the bulb, you cannot take it off. You can unscrew it right away, right? Because it's very hot. Why is it hot? What is giving the light? Because when it's off, you see a little filament. When it's transparent, right? And not frosty, it's transparent bulb. You can see there's a little filament inside, a little black filament, or it's a metal. That electricity goes through that metal. A lot of electricity goes through that little tiny metal, and it makes the metal what? First red hot, but then immediately white hot. It gets so hot that it becomes white hot, and that's the white that we see. In other words, photons are being emitted, but it's hot. And part of that heat is trapped by the glass. And so we cannot touch what is known as an incandescent bulb while it's on because it's very hot. Okay? So the core of the Earth, a ball of iron that is white hot, solid. And that white hot is keeping the mantle flowing, liquid, all right? And then, but we at the surface, at the crust here, are far enough away from that iron core to be a little cooler to have liquid water and the possibility of life. Okay? All this has now, today, an organic, scientific explanation. That doesn't need a magician to be creating and destroying matter and energy. All right, so the Earth is an open system. There is an increasing complexity. What is the increasing complexity? Basically the 7 steps of life, because life is super ordered. From gas, the methane, the primitive atmosphere, what was it? Methane, ammonia, water vapor, and hydrogen gas. Those are gases. What are the gas laws? What is the distribution? What is the gas in here? What do we call the gas in here? Air, right? Told you it's got nitrogen, it's got oxygen, it's got a little bit of CO2 and other, mostly nitrogen, but it's gas. So is all the nitrogen over here?
No.
Or over there? Where is it? The gas laws. The gas takes up the size of the container. Okay, so it's random molecules moving around when in gas. So we had hydrogen, methane, oxygen, and ammonia gas, just in the atmosphere, loosely held in an atmospheric way. In other words, around the Earth. Why didn't it fly out into space? Because those gases have molecules. Those molecules have protons and neutrons, which have mass, which react to gravity, the gravity of the Earth, the gravity of the iron core. All right, so that keeps the atmosphere in place, but still gases. So these gases are just flying around. From that to get life, the life that we see today, 4.5 billion years ago, was a tremendous ordering event. But to maintain the dynamic system, if we do order on one side, we have to increase disorder somewhere else. Disorder meaning randomness. The randomness is going to come, by and large, by the heat diffusion. By the heat diffusion that has been going on Earth, out into the universe, dissipating into the universe, contributing to the entropy, to the energy that is unavailable for work. So there's a balance there in the open system. Okay, the increasing complexity by forming life must also increase the entropy, the second law of thermodynamics, somewhere else in the universe, which is the dissipated heat that we have been giving off. So organic life develops and evolves on Earth. Entropy also increases elsewhere. Okay, I wanted to look at examples of life from unicellular, from a single cell to multicellular and beyond, but it's almost 12:45, so I'm going to stop here. Let me see, there was a message. Any questions or comments? No, people are good. Yeah. I'm sorry, you had a question.
Yes. Off topic. I'm Fisher. So my question is, I understand how our energy moves.
Mm-hmm.
We have a saying as fishermen that the tide is affected by the moon.
Yes.
High tide, low tide is affected by the moon.
Yes.
And we see that, and there's evidence of it.
Yes.
How does that happen? Because
Very good
all
question.
the moon does is reflect the rays of the sun.
Yes, but not all, right? So the moon is an opaque body, meaning that it has mass. If it has mass, it exerts gravity, because the moon also has its core, and the moon itself has mass, has matter. All matter exerts gravity. And so what we see of the tide is actually the gravity of the moon pulling on the water of the Earth, and it makes a bump of water. So here's the
Because
Earth.
waves do get higher.
Yes, well, waves are one thing, but tide is different. You can have waves. Waves are mostly the wind blowing on the surface, okay? But the tide, the rise and the fall of the tide, once a day in the Atlantic, twice a day in the Pacific, because it's much bigger, is the pull of the moon on the water of the ocean. So it makes a bump on the water. The water of the ocean wants to go up to the moon because it's being pulled by the moon. But of course, the pull of the moon is not stronger than the pull of the Earth. So the water doesn't actually leave Earth, because at some point Earth says, no, no, you belong to me, get back down here. But anyway, as the moon rotates or translates around the Earth, it's pulling that bump. It's pulling that bump of water, and the bump of water is unobstructed until it hits land. And when it hits land, you get the crest of the bump is a high tide. And then there is a slump because the moon is going to come around again and pick up another bump over here at the other shore. But in between shores, there is a trough, which is the low tide. And so tides is water going from bump to bump, from shore to shore, being pulled by the moon around the Earth. Interesting, huh?
Yeah, right.
That the tide is actually the moon pulling on our water, but the Earth not letting it go totally.
Yeah, yeah.
Okay, let me stop the recording here. And